Good morning and welcome to class 4. We're continuing chapter 3 in which we will examine the cell structures of eukaryotes. We spent last lecture talking about prokaryotes. We're going to talk very briefly, extremely briefly, about archaea at the beginning of this lecture, and then we're going to delve right into eukaryotes. I'll spend a lot of time contrasting eukaryotes and prokaryotes. So archaea were actually thought to be bacteria for a long time, up until the 1970s when Carl Wolfs was able to sequence the 16S ribosomal RNA gene of these so-called bacteria and found that they actually belonged in a class of their own, which he called archaea. Very similar to bacteria, archaea have structures such as glycolax, flagella, and fimbriae. These are all found in bacteria, and we talked about these in the last lecture. And both archaea and bacteria have cytoplasm. The cytoplasm, though, in archaea contains 70 S ribosomes. These are closer to eukaryotic ribosomal proteins. This will become important when we talk about the translation of DNA into the translation of RNA into proteins in a later lecture. Some other differences included the fact that we have not identified any pili in archaea. We have identified another structure called a hami. These are present in archaea and not found in bacteria. Their main role is to help with attachment of archaea to surfaces. Another difference is that the cell wall in archaea are made of polysaccharides, not peptidoglycan, and the cytoplasmic membrane has a different lipid bilayer. In some cases, it's a single lipid layer, but in most cases, it's a lipid bilayer. But this lipid bilayer is made out of ether linkages, not ester linkages. And I just wanted to point out that ether linkage here. So here's a lipid, a phospholipid. Here's the phosphate group. Here's the fatty acids. This is our L-glycerol. And you can see that there are ether linkages that hold the L-glycerol onto the fatty acid branched chains, as opposed to the ester linkages of eukaryotic or bacterial phospholipids. This makes the archaea phospholipid much more stable in extreme conditions like extreme heat. And so one of the places that we often find archaea are in extreme conditions that have extremely high temperatures. And these branched tails that are different from the unbranched tails of the eukaryotic and bacterial phospholipid help in that stability as well. There aren't a ton of archaea that are associated with humans or human disease. There is one archaea that is associated with disease, and that is the Methanobrevibacteria oralis. This is an archaea that doesn't seem to cause disease on its own, but it does seem to promote the growth of phosphate-reducing bacteria that are absent in healthy controls. So this role may be to actually promote the growth of pathogenic bacteria, but not to cause disease itself. These are confocal micrographs of the bacteria and archaea in subgingival plaques from a patient with severe periodontitis. This is the bright field micrograph just showing the population of bacteria and archaea. And in this, we have the fluorescent in situ hybridization in which you can identify, you are able to pick out individual archaea in this background of, individual archaea in the background of bacteria. So now we're going to move into the eukaryotic cell, and we're going to compare and contrast bacteria to eukaryotes. So let's start out with our bacterial cell. Just a reminder, this is a fairly simple cell that doesn't have a nuclear membrane, and so proteins and ribosomes and everything else are able to freely interact with nucleic acids. There aren't any membrane-bound organelles in a bacteria, and it's a fairly simple structure compared to the eukaryotic cell. So today we're going to spend most of our time talking about the eukaryotic cell and these different membrane-bound organelles that are found in the eukaryotic cell. Specifically, we're going to be talking about the nucleus and the endoplasmic reticulum and several other of these structures, and talking about how proteins and other things are shuttled from the inside to the outside of the cell and vice versa. Eukaryotic cells come in lots of shapes and sizes. The bacteria we tended to see rods or cocci. Archaea tend to have lots of different shapes because their cell walls are slightly different than bacteria, but in eukaryotes, we have a wide variety of different shapes and sizes of cells. So for example, here we have parasites, Toxoplasma gondii, that are actually able to reside inside larger eukaryotic cells. Here we have sea urchin sperm, and you can see that they're basically a head with a tail that looks very similar to sperm found in other animals. We have an algae over here that has a very distinctive um, shape to it. This is a plant cell, and the plant cells have very rigid cell walls that help the plants maintain a shape. And then here we have neurons. These are all eukaryotic cells. They have different sizes, they have different shapes, they have different functions. You can imagine that a neuron cell is very different and has a very different function than a plant cell, but they are all considered eukaryotic cells. And the reason for that is that they all have a nucleus, this true nucleus in which the DNA is contained within a membrane. These membranes are really important in eukaryotic cells for several reasons. The first is that they increase the surface area without increasing the size of the cell. So for example, here we have the endoplasmic reticulum, and you can see that the endoplasmic reticulum membrane has several folds to it, and this increases the surface area for protein synthesis. We'll talk about the role of the endoplasmic reticulum in just a minute. But in this case, we have increased places for protein synthesis to occur without increasing or changing the shape or the size of the cell. In other cases, we are able to sequester away harmful substances from the rest of the cells. So for example, here we have the peroxisome. The role of the peroxisome is to store things like hydrogen peroxide that are used to break down microbes and other things that may harm the cell. Because that hydrogen peroxide is sequestered away or kept away from the rest of the cell by this membrane, the DNA of the cell that's located in the nucleus here is not damaged by the hydrogen peroxide that's found in the peroxisome. I wanted to show this plant cell one more time and show it just a little bit closer to point out a couple of things. One is that many eukaryotic cells share similar membrane-bound organelles. So for example, almost all eukaryotic cells have endoplasmic reticulum. They all have ribosomes. Almost all of them they all have a nucleus. Most of them have nucleoli. Most of them have vacuoles and mitochondria. Unlike other organisms, other eukaryotic cells, so not all eukaryotic cells have plastids. We'll find in other eukaryotes, such as parasites, that there may be other organelles that are found in, for example, the kinetoplast parasites that aren't found in human host cells. And so although most eukaryotes share most of their membrane-bound organelles, there are some organelles that are unique to eukaryotes that we're able to use as targets for drugs. The eukaryotic cytoplasmic membrane is found in all eukaryotic cells. This is a fluid mosaic of phospholipids and proteins. There are a number of recognition molecules, enzymes, receptors, carriers, and channels that are found in the cytoplasmic membrane. And the cytoplasmic membrane may have sterols in certain types of cells, for example, in the fungi, and those make nice targets for antimicrobial drugs. So I want you to take just a minute here and pause the show and think for a minute about why many antimicrobial drugs target bacterial cell walls. Why aren't there many drugs that act against eukaryotic cytoplasmic membranes? You might want to take a minute, look at your book, discuss with a friend that you may be listening to this with. Okay, go off and discuss. Hopefully you find a friend that you can discuss this with. So just like prokaryotes, eukaryotes need ways to move proteins and things that they need from the outside to the inside or the inside to the outside. When eukaryotes move things from the outside to the inside, they often do this by endocytosis, endo referring to bringing in, cyto referring to the cell. So this is the mechanism for bringing things into the cell. Here we have a pseudopod, and you can see that there's a eukaryotic cell with the pseudopod sticking out. It wraps the pseudopod, this eukaryote wraps the pseudopod around this organism and completely engulfs it, and then this pseudopod fuses and engulfs this microbe so that they can, this eukaryotic cell can digest this microbe and extract its nutrients. There's a great little video on YouTube showing this process of a pseudopod. I'd strongly recommend that you take a look at it. There are certain types of active transport processes that are found only in eukaryotes. These are processes involved in endocytosis and exocytosis that aren't found in bacteria. So for example, endocytosis includes phagocytosis and pinocytosis. Phagocytosis in particular is used by immune cells to bring microbes into the cell so that they can break them apart and present small parts of them to other immune cells. In this case, the substances are surrounded by pseudopods. So as you can see up here, we have pseudopods and brought into the cell. Phagocytosis involves solid substances. Pinocytosis involves liquids. And then we have exocytosis. This is moving things from the inside of the cell to the outside of the cell. And vesicles containing substances are fused with the cytoplasmic membrane, dumping their contents to the outside. This is a great way for cells to get rid of their waste. Not everything that the cell brings in is really good for the cell. They don't want all of the substances that they bring in. They produce waste, just like we produce waste. And so they get rid of their waste by exocytosis. Again, this YouTube video is a great reference for that. Just wanted to give you a close-up of a pseudopod engulfing a eukaryote. So, another microbe. So here we have a microbe, and this microbe has been surrounded by the pseudopod. The pseudopod has now fused the rest of the eukaryote, and this creates a vacuole, and the vacuole can now digest this microbe and break it down into parts that it can use. So now we're going to move on to the way that microbes, eukaryotes in particular, move. In our last class, in our last lecture, we discussed bacterial flagella. We're going to contrast that right now with eukaryotic flagella. So here we have a picture of bacterial flagella on the left and eukaryotic flagella on the right. And just from this illustration, you can see that there are several differences. So for example, where we had the rings that were part of the bacterial flagella, in this case, this is a gram-negative bacteria, so there are 4 rings in this flagellal body. Those are absent in the eukaryotic flagella. The structure of the flagella is different. So here we have a tube of proteins, basically a single tube of proteins that has been made and pushed out of the bacterial cell wall. This is different in the eukaryotic cell, in which the flagella are made of these multiple tubules. Seen here, these microtubules that are stretched from this part, the basal body, and actually through the basal body of the flagella all the way out through the part of the flagella that sticks out of the cell in the side in the exterior of the cell. Bacterial flagella do not have these microtubules. These are unique to eukaryotic flagella. There are some really nice videos on eukaryotic flagella and cilia that you should take a minute to watch. In particular, the video shows paramecia that use cilia to move, and I really like the video that we have here because it also shows stentor using cilia to eat green algae. So definitely take a look at that video. In addition to flagella, eukaryotic cells can use the cytoskeleton to move. The cytoskeleton is a bunch of microfilaments or tubes that are underneath the cell wall of the eukaryote, and the eukaryote can actually contract and expand these microtubules to be able to move. There's a nice video of Listeria monocytogenesis, which is a bacteria, and the bacteria uses the actin that's found in the eukaryote to move. So take a look at that video as well. So now we're going to move on to the non-membranous and the membranous organelles of the cells. As we said in prokaryotes, there are, as we said earlier, there are some things that are found in all cells. So for example, ribosomes are used for protein synthesis, and they're found both in prokaryotes and in eukaryotes. And these ribosomes are found in all prokaryotes and all eukaryotes. And there's a little column here that's missing, that's the archaea, because we tend not to talk about those in this class because they aren't actually often associated with humans, but archaea also have ribosomes. The cytoskeleton is present in most bacteria, but not all of them, but the cytoskeleton is present in all eukaryotes, and this helps give shape to the cell. There are some very distinct differences between bacteria and eukaryotes that we're going to delve into. In particular, look at this entire list. These are all things that are not found in bacteria that are found in eukaryotes. So for example, eukaryotes have centrosomes, nuclei, endoplasmic reticulum, Golgi bodies, lysosomes, peroxisomes, vacuoles, vesicles, mitochondria, and chloroplasts. None of these are found in bacteria. Interestingly, some of these are actually originated from bacteria. So for example, we know for sure now that the mitochondria and the chloroplasts actually started out as bacteria that were engulfed by other microbes and became the mitochondria and the chloroplasts of eukaryotes. Now, the eukaryotic nucleus really is one of the identifying characteristics of a eukaryote. So here we have the eukaryotic nucleus. This is a TEM to show you what the nucleus looks like in an actual cell. This is a drawing of a cell, just giving you a different view of the nucleus. A couple of structures that I want to point out. The nucleolus is where RNA synthesis occurs. We're going to talk in a couple of lectures about the transcription of DNA into RNA and then the translation of RNA into protein. In eukaryotes, these processes are separated by this nuclear membrane in which the DNA can be transcribed into RNA, and then the RNA is actually processed and proteins are processed outside of the nucleus. The nucleus is surrounded by a lipid bilayer, and it's not just one lipid bilayer, it's actually two phospholipid bilayers. The nuclear membrane has a number of different pores. These pores allow movement of molecules, large molecules that can't diffuse passively between these two phospholipid bilayers, to move from the inside of the cell to the outside of the cell. And then attached to the nuclear membrane is the rough endoplasmic reticulum. We'll talk about the rough endoplasmic reticulum and the smooth endoplasmic reticulum in just a minute, but I wanted to show you just how tightly associated the ER, the endoplasmic reticulum, is to the cell membrane, the nuclear membrane. This is taking another look at the nuclear membrane. So here we have the outer membrane of the nucleus and the inner membrane of the nucleus. The nucleolus, as I said, is where RNA is actually transcribed from the DNA, so that's an important part of the cell. It's not separated from the rest of the nucleus by a membrane. It's actually free-floating in here, and that allows proteins and nucleic acids to move freely from the rest of the nucleus into the nucleolus. So there's no separation really of space, it's just that the nucleolus is tightly bound nucleic acids. There's a lot of chromatin found in the nucleus. The nuclear envelope itself, as I said, has the inner membrane and the outer membrane. These are two phospholipid bilayers, and they're closely associated with the endoplasmic reticulum. And here you can see that the endoplasmic reticulum is actually branching off from the nuclear membrane. And again, there are nuclear pore complexes that allow larger proteins to move in and out of the cell. Now, important to all this is the fact that DNA is transcribed into mRNA, which is then translated into protein. In the prokaryote, the bacteria that we talked about in our last lecture, there was no structure that was separating all of these processes from happening. DNA could be transcribed into mRNA and then could be translated into protein, all within the same structure, so it happened very quickly, and there was no separation of these processes. But in eukaryotes, we have DNA that is transcribed into mRNA within the eukaryotic nucleus, but then the mRNA is shuttled out and translated into protein in a different organelle. So that requires a movement of mRNA in and out of the nucleus, which also means that this translation of the protein is separated from the transcription of the RNA to the from the DNA to the RNA. The endoplasmic reticulum is another organelle that is actually tightly associated with the nuclear membrane, and this is where protein synthesis actually occurs. There are two types of endoplasmic reticulum. The smooth endoplasmic reticulum is the part where lipid synthesis and transport occurs, and then the rough endoplasmic reticulum seen here is called the rough endoplasmic reticulum because it looks rough, and that is because there are a ton of ribosomes that are tightly associated with the endoplasmic reticulum. Each of these red spots here is a ribosome, and you can see that they just completely cover this endoplasmic reticulum. Those ribosomes are used for making proteins. So this is where the translation of RNA into protein occurs. And just a reminder that translation of RNA into protein is separated from the DNA that's located here in the nucleus. I wanted to point out these little vesicles that are blubbing off the endoplasmic reticulum. These vesicles will be shuttled to the Golgi apparatus. The Golgi apparatus is an apparatus that's used to give the proteins their shape and to modify them in different ways. And it's actually a fairly complicated structure. And so I wanted to show you a video of how things actually move in and out of the Golgi body. So unfortunately, you can't hear the sound coming from this video with this particular type of recording going on, so I'm just going to have to tell you what's going on. So this is a video. The link is in the PowerPoint, and this video shows you the movement of proteins within these vesicles through the Golgi apparatus. So here we have the Golgi apparatus, and you can see that it has several different portions. This is the cisterna, which is closest to the endoplasmic reticulum. This is the medial and transcisternae, and this is the trans-Golgi network. Other key players are the proteins being transported, and then Golgi enzymes that can modify these proteins. They can modify the shape and other parts of the proteins. So here we have the endoplasmic reticulum, and vesicles are budding off the endoplasmic reticulum and fusing together. These fused vesicles form the cisterna. The protein moves through these sacs, and the Golgi enzymes modify these proteins within the Golgi apparatus. The modifications provide the signal that tells the protein where it's going to go. So the proteins actually go through the cyst, the medial, and then the trans. So here we have some vesicles that are budding off the ER. They're forming the new cisterna. These medial cisterna are now being pushed here towards the trans cisterna, and this trans cisterna now forms blebs. Form blebs in which the proteins have separated based on their modifications into these different blebs. And these different blebs will turn into vesicles that will now move throughout the cell. So here we have the trans-Golgi network budding off into vesicles, and these individual vesicles will move to their different locations. Some of them will go to the lysosome, which is the digestive vesicle in the cell. Others will be shuttled out of the cell as proteins that will interact with things outside of the cell. As you can see, that's a fairly difficult, that would be fairly difficult to show in a PowerPoint. So I'm glad I was able to pull that up. But my voiceover is not nearly as good as the voiceover that's used for the actual video, so you should check out this video. Start it at about 1 minute and 25 seconds into the video to see the part that we just saw. So there's a nice video on YouTube showing endo and exocytosis, and I would highly recommend watching this video as well. Basically, endocytosis is the cell membrane folding in around a microbe or something that the cell wants. In this particular case, this is phagocytosis, so this cell membrane is wrapping itself around this microbe. The microbe is now sequestered away from the rest of the cell in this food vesicle, a phagosome. The phagosome fuses with a lysosome. This lysosome contains a bunch of things that can destroy the microbe, including a change in pH and different salts and enzymes that are really good at breaking down the microbial cell wall. So this microbe is now broken down into little bits and pieces in the phagolysosome. The phagolysosome is what this lysosome is called when once the vesicle has fused to the lysosome. This is now phagolysosome. The phagolysosome is used to break down the microbe. The microbe then, once it's broken down, is either shuttled out or bits and pieces of it are presented on the surface of the cell. So this is endocytosis, in which the microbe is being brought in, and then exocytosis is this vesicle fusing with the cell membrane and then pushing the contents out. This can also occur, exocytosis can also occur from the Golgi body. So as I showed you before, vesicles bleb off of the Golgi body, and these vesicles contain different proteins. These secretory vesicles can then fuse with the cell membrane and release the contents of the cell membrane to the outside of the cell. So that is so now we've covered the nucleus, the endoplasmic reticulum. You should be able to differentiate between the smooth endoplasmic reticulum and the rough endoplasmic reticulum. We talked about the Golgi apparatus, and we've talked about endocytosis, which is seen here, and exocytosis in two different manners, both from phagolysosomes using exocytosis to get rid of microbial contents, and then also the Golgi apparatus shuttling proteins that are important for the cell to maybe signal to other cells or interact with other cells by pushing it out of the protein, out of the cell. So now we're going to move on to the mitochondria. Mitochondria are fascinating, in part because this is the powerhouse of the cell. We talked about this molecule ATP that is so very important for providing an energy source for cells. A lot of ATP is actually made here in the mitochondria. And we talked about the fact that membranes can increase the surface area that allows for more processes, more reactions to occur because there's more surface area for these reactions to occur. You can see in the mitochondria there are a bunch of folds. These are the crista. All of these folds provide surface areas for more and more and more energy to be made. Also, the mitochondria has their own ribosomes. This was important for identifying the origin of the mitochondria. So there is a fabulous scientist, Lynn Margulis, who was instrumental in identifying the origin of the mitochondria as being bacteria. So it turns out that one cell actually engulfed another cell, and that second cell became the mitochondria for the first cell. So this is the origin of mitochondria, but this is also the origin of chloroplasts. And they've been able to identify that mitochondria are able to produce ATP for the eukaryotic cell. They are now essential for eukaryotic cells. There are very few eukaryotic cells that do not have mitochondria. So this has really become an organelle that the cell relies on. And the same is true for chloroplasts for algae and other eukaryotes that are able to convert light into energy. The chloroplast for those cells has become essential, and if you remove the chloroplast, then these cells actually die. And another important component is that if you take the mitochondria or if you take the chloroplast out of the eukaryotic cell, the mitochondria and the chloroplast do not survive on their own either. So this is truly an endosymbiosis where the mitochondria or the chloroplast have to have the eukaryotic cell to survive, and the eukaryotic cell has to have these organelles to survive. So what does endosymbiosis actually look like? So this is the current theory that's going around. There was once a proto-eukaryote. It kind of looked like a eukaryote. It kind of had a DNA that was sequestered away, but there were no membranes around the DNA. At some point, either one cell engulfed another, and that created the nucleus, or infoldings of the plasma membrane with the ancestral prokaryote gave rise to endomembrane components, including the nucleus and the endoplasmic reticulum. Then at some point, and remember that bacteria are, I think the latest estimate is 1.5 billion years old, and eukaryotes are several hundred million years old. So I'm not talking about something that happened over a couple of years. I'm talking about something that happened over thousands of years. So this this pre-eukaryotic cell or a eukaryotic cell eventually absorbed an aerobic bacterium, and instead of killing the aerobic bacterium, which is what happens so frequently when a bacterium is taken in by a eukaryote, instead of destroying this aerobic bacteria, the cell actually turned it into the powerhouse for the eukaryotic cell, and that became the mitochondria. And these cells that had a mitochondria then later on, perhaps thousands of years, who knows, later on absorbed a photosynthetic bacteria, and that photosynthetic bacteria is now the chloroplast for modern photosynthetic eukaryotes. So this is basically what endosymbiosis looks like. So now we've covered most of the membrane-contained organelles that are found in most eukaryotes. I wanted to talk about some of the specific proteins and other things that are found in eukaryotic cells, including the ribosome. The ribosome is really important for transcribing RNA into protein. And we talked about this very briefly just a few minutes ago when I talked about the making of proteins in the endoplasmic reticulum. And I showed you those ribosomes that were covering the rough endoplasmic reticulum. This is what the ribosomes look like when you look at them a little bit closer. And you can see that there's a spot in the ribosome for RNA to come into the ribosome. And then there are these particular proteins that will add an amino acid to a growing polypeptide chain based on the sequence of nucleotides that are found in the RNA. And if all of this sounds confusing right now, we're going to go into much greater detail on this when we talk about, when we get to the chapter on genetics. But the particular thing that's important about ribosomes is that ribosomes give a safe place for translation to occur. And it's separated from the DNA. The cytoplasm and the endoplasmic reticulum are used for translation of this RNA into protein, so that's where the ribosomes are located. The instructions for this RNA are encoded in the DNA. So we're going to show in a couple of lectures how our DNA is transcribed into RNA. And then the materials for RNA, the nucleotides for RNA, the amino acid modifiers and whatnot, are found in the endoplasmic reticulum and the cytoplasm. And that is one of the benefits of having membrane-bound organelles, is that you can actually contain or concentrate your amino acids. You can concentrate your nucleotides that are specific for a particular reaction. In this case, the nucleotides for RNA and DNA are found in the nucleus, and then the amino acids are found in the endoplasmic reticulum and some in the cytoplasm. The endoplasmic reticulum is also really great for getting the product where it's needed. We talked about the modification of these proteins We talked about the synthesis of these proteins in the endoplasmic reticulum, and then the modification in the Golgi apparatus, and those modifications indicate to the cell where those particular proteins need to go. And then for proteins that are translated and turn out to be bad proteins, then the endoplasmic reticulum and the Golgi apparatus are really good at getting rid of those, acting as a garbage can to use exocytosis to get rid of those proteins, shuttle them out of the cell so that they can't damage the cell. Okay, so now we're coming back to the nucleus, and the reason I wanted to come back to the nucleus is I wanted to focus in on this nuclear envelope. So here we have the nuclear envelope. It contains nuclear pore complexes, and it's tightly associated with this endoplasmic reticulum. The endoplasmic, the eukaryotic nucleus is really important because it separates the replication of the DNA and the RNA from the production of the protein. The nuclear membrane serves as a barrier between DNA and the rest of the cell. It's fused to the endoplasmic reticulum. There are small proteins and metabolites that can traverse through these nuclear pores, and that's important because those nucleotides and the small proteins and the metabolites are important for driving the machinery that transcribes and translates replicates DNA and transcribes DNA into RNA. And then the nucleopores also export mRNAs into the cytoplasm. So as I said, there are some proteins that are actually translated in the cytoplasm. These nucleopores can shuttle mRNA messages that have the right sequences out of the nucleus and into the cytoplasm where they can be translated. The nucleolus seen here is the site of ribosomal RNA transcription and processing and ribosome assembly. So this is basically the ribosome factory. So let's take a look at the closer look at the nucleus. So here's the nuclear envelope and the pores, and here you can see individual pores. This is a TEM. So it's a nice, really close look at the nuclear membrane. Here we see the cytoplasm in here. This is the nucleus in here. So the DNA would be in here. This is the cytoplasm outside of the nucleus. And this is the nuclear envelope. And you can see that at the nuclear envelope, there are a number of pores. And there are these pieces of RNA that are trying to get through these pores. This is taking a closer look at the rough endoplasmic reticulum and the ribosomes. Here you can see the individual dots. Each of these individual dots is a ribosome. Ribosomes are actually fairly large proteins, and so they are one of the proteins that were the earliest to be described in their shape and their sizes and whatnot. This rough endoplasmic reticulum, the ribosomes are attached to the endoplasmic reticulum and gives it a particular rough-looking shape. It's contiguous with the nuclear membrane, so it's actually bound to the nuclear membrane. The cells specialize in making proteins. Cells that specialize in making proteins have a ton of rough endoplasmic reticulum. When we get to B cells, we'll talk about B cells making a whole lot of antibody, and because they're making a ton of antibodies, they have a lot of rough endoplasmic reticulum because they're basically protein-making cells, right? Cells specializing in making proteins have more rough endoplasmic reticulum. Cells producing lipids and fats and steroid hormones actually have more smooth endoplasmic reticulum. Modification and folding occur within the rough endoplasmic reticulum, and then misfolded proteins are targeted for destruction. The ribosomes take polypeptides and amino acids from the cytosol and translate mRNA into protein. I showed that just a minute ago. Can do a ton of them at a time. These large proteins can make, really large proteins can take hours to be produced, like a 300,000 amino acid muscle protein called Titan takes hours to be produced, but most proteins are made really quickly, and that's because 200 amino acids can be joined per minute. Whoa, sorry about that. There can be up to 10 million ribosomes per cell, so you can see each one of these dots. There can be up to 10 million of these per cell, and they exist both in free form floating around in the cytosol, but then also bound to the endoplasmic reticulum. Here's another look at the endoplasmic reticulum. Here's the endoplasmic reticulum here, smooth endoplasmic reticulum here, rough endoplasmic reticulum here, because we do have the ribosomes there, and you can see that it's actually bound to the nuclear membrane. The smooth endoplasmic reticulum makes lipids. So, for example, in the liver, there's a whole lot of smooth endoplasmic reticulum that enables the breakdown of glycogen to glucose. And then smooth endoplasmic reticulum is also used to make steroid hormones such as adrenal cortex and in the endocrine glands. Another role is to detoxify organic chemicals to safer water-soluble products. So, for example, in the liver, the smooth endoplasmic reticulum is used to detoxify ethanol and barbiturates. Another important organelle is this lysosome. The lysosome is really important for breaking down bacteria. So obviously this is something that is not found in bacteria, it's found in eukaryotes. There can be up to 50 different enzymes found in these lysosomes. And these different enzymes are used to break down proteins, DNA, RNA, polysaccharides, lipids, everything that you can find in a microbial cell. There are a ton of enzymes in the lysosome that are used to break down those cells. In addition, the lysosome has a very low pH, so often the lysosome can have a pH of about 5, whereas the rest of the cytoplasm has a pH of about 7.2. And remember that 7 is considered neutral, 5 is more acidic. Another organelle that's found in eukaryotes that's not found in bacteria are these peroxisomes, and I talked about these earlier. These peroxisomes have a number of molecules that are good for breaking down cells. They produce hydrogen peroxide, which then makes a catalase to break down hydrogen peroxide into water and oxygen, and they can break down they can break down long chain fatty acids by oxidation and break down uric acid and amino acids. Oops. These peroxisomes are also used to break down fatty acids to create energy. So, for example, there are enzymes in the peroxisomes that can break down fatty acids and turn them into carbohydrates or sugars. There can be different sizes of peroxisomes. Here we're showing a small peroxisome relative to this lysosome, but they actually come in lots of different sizes. And they can increase in number. So a cell can produce more peroxisomes when the cell is exposed to a challenge like alcohol. And in this case, if the cell is challenged with something like alcohol, the peroxisomes can be increased to occupy fully half of the cell in order to break down that molecule into something that's less toxic for the cell. Because they're really good at breaking down things into something to detoxify them, there are a ton of peroxisomes in liver cells, because remember the function of the liver is to detoxify blood. Whoops. Okay, so there are a couple of videos you can take a look at to examine exocytosis and endocytosis a little bit closer. One of the last few things that I wanted to talk about is cell division. So we'll take a look at cell division a little bit closer when we get to genetics of different cells, but I wanted to give you a brief overview. So in eukaryotes, in prokaryotes, in bacteria, cell division was binary, right? One individual cell duplicates its DNA and then produces a daughter cell. So one cell makes two cells make 4 cells, et cetera, et cetera. This is different in eukaryotes. Eukaryotes undergo mitosis and meiosis, and the difference basically is the replication of the DNA. So here we're showing mitosis in a cell in which a cell is replicating its DNA. The DNA is separated out onto different poles, and then cells, individual cells are made. In each one of these has a copy of the DNA in it. And then this just shows the different phases of DNA replication. During prophase, the nuclear envelope fragments and rebuilds during telophase. So here's prophase. The nucleus is actually broken down, and you can see that here there's no nucleus here in metaphase or in anaphase, and then here in telophase, the nucleus, that organelle that's so important for separating DNA from the rest of the cell, in telophase, that nucleus is reformed. Let's see, we talked about the mitochondria, which is the powerhouse of the cell, and the chloroplast, which turns energy, it turns light into energy, so it turns energy from the sun into ATP and reduces NADPH in light reactions, and then also turns ATP and carbon dioxide and turns them into sugar. The precursor was cyanobacteria, which uses thylakoids and stroma similar to bacterial cytoplasm, and importantly for identifying chloroplasts, they have their own DNA and ribosomes. Okay, so when you are reviewing these slides, to think about what's important, one of the things that I want you to focus on is the difference between eukaryotic flagella and bacterial flagella. And another thing that I want you to think about is differentiating flagella from cilia. So cilia, it turns out, so flagella are used to move, and bacterial flagella are used to move, and then cilia are found in eukaryotes but are not found in prokaryotes, and the cilia are used to move as well. Okay, that's about it. I will have questions available on Blackboard for you to answer. Feel free to go back through the slides again. In addition to having my voiceover, I'm going to also upload the individual slides as a PowerPoint or as a PDF that you can take a look at without having to listen to my voice. Don't forget to look at those videos. Those are some really great videos for wrapping your head around the different processes, the different things that I showed you in today's lecture. That's it, guys. Have a great day.